Hello, everybody. My name is Yannick. Thank you for joining in. We will still wait 5 more minutes and start 4:05, so everyone has the chance to join the webinar. And we will talk in 5 minutes. Thank you very much.
Yes.
Just 1 more minute, and then we will start the webinar. Thank you for your patience. So, 4:05. Let's start the webinar. Hello and welcome everybody to our webinar of the Permeability series, in vitro tools for predicting biopharmaceutical performance of drugs in vivo. My name is Yannick and I will be your host for this webinar today. On today's agenda, we will take a closer look on tools and barriers that help you to investigate the permeability of drugs. From the basic question why to use in vitro tools to predict in vivo performances, to the explanation of an important indicator, the apparent permeability, and concluding with practical examples of experiment setups. We are very happy to again have Max as our presenter for this live webinar. Max has a very impressive academia vita and can therefore be called a total expert in pharmaceutics and especially in drug formulation and permeability. Next to that, Max is also a freelancing scientific consultant for Inome in Germany, the company which manufactures and commercializes PermaPad. Max co-invented PermaPad at the Southern Denmark University where he used to work. Before we start with the webinar, a few things regarding the organization. We have opened a Q&A window on your right side where you can type in your questions. We will answer them at the end of the presentation and will provide also the slides and the link to the recorded webinar at the end of the presentation. Without further ado, I give the word to Max. Enjoy the presentation and see you at the Q&A session again.
Thank you, Nick, for the nice introduction. I will now share my screen. Okay. I hope the presentation is visible.
Now.
Okay. I hope that the presentation is visible now.
Yes.
Yes. Okay. Perfect. So today I will present to you. I will try to present to you why we believe that in vitro tools are extremely important for the future of pharmaceutics and in the context of predicting biopharmaceutical performance of drug in vivo. The outline of the presentation of today, I will start with contextualizing why PermePad and why in vitro tools are extremely important and for predicting in vivo drug performances. I will go through some concepts related to the upper permeability that were actually discussed in the previous webinar as a refreshment. And then I will discuss which are the most utilized commercially available artificial in vitro barriers available in the market. And then I will make some example of their applicability. But why we have decided to create a series on permeability? Well, the reason are several. The first one is that pharmaceutical research in general is still based extensively of animal experiments. And this opens several questions, ethical, but also in terms of scientific question. Are we doing a correct work in developing a drug? Definitely there are some problem in understanding the permeability as a concept. It is a concept that often is misunderstood or misinterpreted. And that's why results sometimes might not match the expectation. In vitro methods also are definitely not implemented enough, especially at industrial level. And but we do believe that in vitro tools like the PermePad can provide can be very predictive in terms of both active ingredients and screening, but also in enabling formulation testing. Besides the ethical issue, there is a cost and time issue in drug development. So this is the timeline frame of standard development of drug that can from drug discovery up to the marketing. And it can take up to 15 to 16 years. And the discovery preclinical testing part actually can take up to 7 to 8 years. So it's a rather long period of time. And it's not, and it's the time where actually animal experiments are are mostly applied. And this is an issue, first because it takes time, but also because it's actually very costly. So proposing new in vitro tools that can be actually, that can provide in vivo predictions of biopharmaceutics performance of drugs can be of extreme utility. That's why, and also having in vitro tools can also provide extremely good and important parameters that can be later used for the transition from in silico to in vivo, or from in vivo to in silico. That means profiling and modeling. And last but not least, the promoting in vitro tools will promote the 3Rs principles. So refining, replacing, and reducing animal testing. And this is a core principle for the European Union from 2010. But still is not yet implemented to the extent it should be. So if we ask ourselves, which are the tools that we can utilize for predicting in vitro bioavailability, the answer is, well, in the past, it was the partition coefficient, octanol water, but that is now considered definitely insufficient for a proper interpretation of the real situation. Then we went through the cell culture system, so the cellular-based barrier model, once for all, the CACO2 cells model. And then we'll end up now on artificial membranes. So definitely the concept of permeability since the 90s in pharmaceutical drug development has grown substantially. The permeability, and this is a repetition from the previous webinar we had, so I apologize for the one that eventually has attended already, but I think it's crucial to have it again. The upper permeability is a measurement, it's a way to measure how fast drugs, active ingredients, can go through a biomimetic barrier over time. This is measured by diffusion studies, where we actually utilize a donor compartment where we place the drug. We have a barrier, cellular-based, non-cellular-based, whatsoever, a biomimetic barrier, and then we have an acceptor medium. And assuming that the fixed laws of diffusion is valid through all the diffusion process, we actually monitor a flux, so an accumulation of mass of the drug over time in the acceptor, and by a simple division from the total drug concentration in the donor, we obtain the permeability. The permeability is normally expressed in centimeters over second. It's a velocity. It tells how fast a molecule will go through this biological barrier. And why this is important? Because, in terms of drug development, because it has been assumed back in 1995 in one of the most cited paper in the pharmaceutics field from Amidon et al., where the biopharmaceutics classification system was introduced, that there should be a very nice correlation between the upper permeability and the fraction absorbed of the drug. So to say, the oral bioavailability. And what is important is that this achievement, this correlation should also be valid if we utilize enabling formulation. So higher permeation or upper permeability with enabling formulation will tell us that we have produced a formulation that is superior in terms of the available formulation in the market. For this type of permeability studies, we have different type of setups. We have the important and the constant is that we have a donor compartment, a barrier, and an acceptor compartment. We can utilize vertical diffusion cells like the classical France cells. We can utilize multi-well plates, and we also can utilize different type of macroscopic diffusion cells like the side-by-side or zinc chambers. And we, of course, can select the barrier we like the most. The diffusion cells has been developed for ex vivo tissue, so tissue from animals. Nowadays, we have a much more potent and robust and reproducible barrier that are actually industrially produced. As I said before, we have two types of biomimetic barriers, the one that are based on cells, so the cellular-based, and the artificial barriers. Which are the advantages and disadvantages? Well, the cellular-based model, like the CACO2, but also other cell lines that we can grow to test permeation through biological cells, they need some cultivating times that can go from 8 to 20 days. They also need a dedicated lab where working need to be performed in a sterile environment. There is high variability in the results in general and poor reproducibility from lab to lab. But of course, if you use cells, then you can also account for the so-called carrier-mediated transport, the active transport. This is important. Well, this is a good question. It is definitely the passive diffusion is by far the most important transport mechanism for the GI tract for any drugs. For some selective substrates, active transport might be interesting, but they are very few. So passive diffusion, as it is a thermodynamic phenomena or a spontaneous phenomena, physical phenomena, this will account for any compounds. So having a proper assessment of passive diffusion and passive permeability will be sufficient in most of the case to get a nice prediction of the bioavailability. The in vitro cellular approach, the standard goal is the CACO2 cells. This is generally based on a 96 well-placed setup, where heterogeneous human epithelial colorectal adenocarcinoma cells are grown on monolayers. The cells possess an apical side and a basolateral side. And as I reported before, what we actually do, we place a drug solution in the apical side, and we account how much drug permeates through the basolateral side over the time. We also have the possibility to inhibit the efflux or efflux pumps that are present on the cells, so we can actually basically account precisely the influx and the efflux of the drug. If the drug, this is relevant if we have a drug that is a substrate for this glycoprotein and transporters, which is not often the case. The most utilized and implemented artificial barrier is called PAMPA. It has been introduced in 1997-98 by Kansi et al. And PAMPA stands for Parallel Artificial Membrane Permeability Assay. The concept is very similar. It is also based on 96 well plates, but in this case, the barrier is a synthetic, is a filter, inert filter that is soaked with a mixture of lipids and organic solvents. The original composition was N-dodecaine and egg lecithin. Now, there are different types of PAMPA that can mimic more the skin, more the blood-brain barrier, more the GI tract, and this is done by changing the lipid composition. It is ideal for high-throughput screening, but this is important to underline that this is, I would define it as a partitioning-based barrier. So the barrier is an homogeneous lipid layer where the drug needs to go through. So you have a partition into a lipid layer, and then we have another partition again in the acceptor water media. So that said, it cannot account for any paracellular transport, which is actually very important when it comes to understanding the permeation. And this barrier is also a thick barrier that has a rather high resistivity. So when we utilize enabling formulation, it has two problems. One, that it cannot really sustain much excipients or some types of excipients, and also the transition is very low, and it might not be comparable to the human kinetics of drug absorption, but it is utilized to some extent at industrial level. This is a table that resumes the different type of PAMPA that has been developed for mimicking the GI tract, the blood-brain barrier, the skin, for example. But they all have the same issue. They all need to be incubated before utilization for 7 to 3, 4 hours, up to actually 15 to 18 hours. So it can also become long. The experiment can actually be prolonged. Pampa has also been, some Pampa barrier has been also tested for the predictability utilizing enabling formulation. And this is a nice example from a work from Beng et al. In 2017, where they have tested this BCS class 4 etoposide, so very poorly soluble and very poorly permeable. And they tried to formulate these compounds utilizing a micellin agent, the TPGS, tocopherol polyethylene glycol. And you can see here in the bottom graph, they have increased the concentration, and then they observed with the Pampa a reduction of the permeation, of the upper permeability. And the reduction starts when the CMC of the TPGS, so the critical micellar concentration, is overpassed. And this is logical because when the CMC is overpassed, we have a solubilization of the drug, and these are lipophilic compounds that will sit inside the muscles, and therefore we have an increasing of solubility. And in this case, the Pampa model will predict a drastic reduction in absorption. Drastic reduction that actually in real life, that is not really present, because when this formulation has been tested in vivo rat permeability studies, the permeability resulted pretty constant. So in this sense, the Pampa was not really predictive in terms of formulation. And another type of barrier that has been introduced lately, back in 2013, is the PermePed. The PermePed is basically a barrier that is composed sort of a sandwich from two low retention layers and a lipid layer in the middle. And it is commercially available also in high throughput screening, so in 96 well plates. And it is two huge advantages in compared to the Pampa, for example, or the CACO2. It is extremely resistant to chemical and mechanical aggression. That means that we can use any excipient we want. The barrier will hold its structure. And it accounts also for paracellular transport. And why it accounts for, as I said, the PermePed comes in different format, different size of pads that can be applied for macroscopic diffusion cells, but also the high throughput screening, multi-well plates. Ideal if we have a robot and if we really want to screen a lot of compounds in a rapid manner. So when we put this barrier, we place it in diffusion cells. Here we have a side-by-side cells or a vertical diffusion cells, and the barrier is hydrated. The barrier is immediately hydrated and spontaneously within the two layers of cellulose, regenerated cellulose, liposomes. A liposomal gel forms spontaneously. Liposomal gel means that we have liposomes that are all packed together. And and this is the picture of how they looks like. So basically, we have a system that reassembles what is happening in cells. So we have the largest surface area for permeation is a phospholipid bilayer, but also we have interspaces between the liposomes where small hydrophilic molecules can go through. And this account gives a very good estimation also of the paracellular transport. And this has been actually presented and proved lately by a work from Erickson et al., a very elegant paper where they actually have utilized PermePed, and they have worked in measuring permeation of both hydrophilic compounds and lipophilic compounds in different tonicity environments. So using two isotonic solution in the acceptor in the donor, or making the solution more hypotonic or isotonic. And the results was that with the PermePed, we could actually get up to 50% deviation and reduction or increment of permeability values according to the tonicity. Why this is important? Because this is exactly what would happen in real life. The cells are also subjected to tonicity environment changes, and they also can have, and also the permeability of drugs through cellular-based testing models is affected by tonicity and needs to be controlled. So another very important thing is that this effect was observed for a hydrophilic compound, small, like caffeine, for example, but it was much less significant for lipophilic compounds, because the lipophilic compounds has the tendency to go through the liposomes and the cells, so promoting the transport is promoted by transcellular pass more than paracellular. And if we, for example, compare the predictability of Pampa methods, this is actually a figure that have been taken from bioscience.com, that is a company that provide measurements for permeability utilizing the Pampa, in this case the Pampa GI. And here they have tested also caffeine, and according to the test with the Pampa technology, the caffeine results a medium-low permeable compounds. But as we all know, the caffeine is actually probably one of the most bioavailable compounds we have in pharmaceutical products. If we tested with the PermePed, actually caffeine is rated as the highest and more permeable compounds, inducing a prediction of 100% bioavailability. That is actually what is happening in real life. And the reason of that is that where the Pampa is a partitioning-based barrier, where the drug needs to go from a water-based environment to a lipophilic environment, here small molecules can go through the paracellular transport and be correctly interpreted in terms of bioavailability. PermePed has also been several studies on the PermePed plates has also been performed to see how this this high throughput screening approach could be predictive for the bioavailability of different compounds. And they have, these are I reported these slides just to show that the volumes that are involved and the size and the volumes that are involved in this kind of assays are very similar to the classical 96 well plates we utilize and the Pampa. And what we have been doing also is not just to test active ingredients, but also to test enabling formulation. And here is the latest results produced by Tsanova et al., where they actually have investigated the role of different environments, donor environments produced by addition of glycerol, polyethylene glycol of different chain length, and on the net permeability, showing that hydrophilic compounds are much more sensitive to changes in the donor compartment than the lipophilic compound hydrocortisone in this case. And actually, it is also possible to discriminate. Here, it's not reported the permeability, but the resistance, that is the opposite of the permeability. And in this case, it is possible also to focus on which of the permeability layers is the most effective or the most impacting of the net permeability. So to say, the unsterile water layers in the acceptor and donor media and the barrier itself. In these other studies, PermePed multi-well plate has been utilized to screen 14 model compounds and to see how the permeability of these compounds could be correlated to the human fraction absorbed and bioavailability in humans. And actually, the correlation they got in this work is actually extremely good, up to the CACU2 cells, basically, indicating that PermePed is really a very nice permeatic barrier, very close to reassemble the logarithmic correlation between fraction absorbed and upper permeability that, in theory, should be present. And the last barrier I want to discuss and to focus on is the so-called regenerated cellulose barrier or the dialysis barrier. In the literature, there are some reports of studies, permeability studies that are performed with this kind of barriers. my understanding is actually that these barriers are not biomimetic barriers. These barriers are size exclusion based barriers. So they actually they can actually discriminate the drug according to their size more than their ability to go through biological barriers. And here I summarized the results of permeability through cellulose, regenerated cellulose I have made during my years, and also from the review from Berben et al. 2018. And the difference is that in the study I presented, the regenerated cellulose has a larger cutoff size between 15 to 20 kilodalton. In these other studies, the cutoff was much smaller. But at the indicatively, in both cases, the permeability measure that you see is rather high. It's a -5, not -6, so 10 times higher than what you generally get with Pampa or Permeped or Caco2. Is basically a correlation 1 over the molecular weight. So that means that the larger are the molecules, the lower is the permeability. And this should not always be the case, because of course we have this is the case only if we don't have a biomimetic lipid layer in the middle. So basically the permeability that can also be expressed as a diffusion coefficient over the thickness, introducing or rewriting this equation considering the Stokes-Einstein equation, will basically tell us that the permeability is 1 over the radius, or in this case, 1 over the molecular weight. So the regenerated cellulose barrier are not biomimetic barrier as such. They are much more permeable than biomimetic barrier, cellular barrier, and they basically rank the drug according to their size, not to their biopharmaceutics properties. In these other studies, we have tested the permaped barrier with liposomal formulation of 2 different drugs, caffeine and caffeine and hydrocortisone. And actually, what we have found out that is if we tested this regenerated cellulose barrier, the permeability predicted was higher, significantly higher for both caffeine and hydrocortisone. But when we introduced a biomimetic barrier, in this case was also permaped, we actually got no difference between the free drug solution and the liposomal dispersion for the caffeine, whereas for the hydrocortisone, a small difference remained. So the formulation was positively influencing the permeation. So the liposomal dispersion was better in terms of local bioavailability or predicted bioavailability for this formulation. And very interestingly, this data actually reassemble perfectly what we have found out with ex vivo cellular tissue from sheep nasal mucosa. So the results are exactly the same. So the permaped in this case was extremely good in predicting in vivo, ex vivo in vivo situation, whereas the cellulose hydrate was absolutely not. So in conclusion, if we just take into account the permaped and the pump and the Caco2, here is just a brief summary, reassemble the properties. So in terms of mechanical resistance and chemical resistance, permaped is basically the best. Reproducibility is also very good. The only thing that Caco2 has of more is that it can account for the active transport. But of course, when it comes to usage of enabling formulation and excipients, then Caco2 is significantly hampered because Caco2 cells are killed by most of the surfactant, and the same as Pampa. Pampa is unstable if we utilize cremophore, solutol, or Triton-X of aggressive surfactants, whereas permaped is absolutely stable and can be tested in any condition. With this, I conclude my presentation of today, inviting you, if you find this presentation interesting, to follow our next webinar that is going to be on the topic in vitro permeability experiment, experimental approaches, and data interpretation in practice. So we will be much more practical in terms of data analysis and understanding what is relevant when we make these types of experiments. And if you have any question, feel free to contact Fabio Crinome, or if you have scientific question, please contact me directly.
Thank you very much, Max, for your great presentation. And I make you a little bit bigger or do it in a duo. So let's start with the Q&A session. I received a few in a private message, so it's not in the general chat. I think we can start with this. Also, for your information, I just shared the slides as a handout in the chat, so you can download it, and you will receive a link to the recording in a few days. Okay, the first question, Max, is what can the knowledge of being able to simulate the paracellular transport with PermePet change?
Well, it changed a lot because, as I mentioned it before, the current setup that are, let's say, synthetic barriers, like a pampa, for example, they cannot account for this. But the paracellular transport is becoming the focus on the paracellular transport is growing a lot. So it is becoming clear that paracellular transport, especially for small hydrophilic compounds, is extremely important. So it's necessary to have this kind of the possibility to account also for this transport mechanism for a proper interpretation or prediction of in vivo biopharmaceutics properties. Yes.
Okay. I hope this answer was clear. If not, please ask again. Now we have one in the chat. I think I switched between them. Is it possible to use PermePet with ethanolic diffusion receptor solutions?
Yes. We are. I don't have data that I can present now, but the first publication on PermePad, the stability was tested up to 15 to 20% ethanol, but this concentration can be raised up to 50% if it's really needed, and it can be extreme. Yeah.
Okay. I think I have a similar question. Is there a surfactant that must be avoided to maintain barrier integrity with KQ2 and Pampa?
Well, some of them I show in the presentation, the most aggressive, Tritonex, Solutol, but for what I know, CACO2 cells are very sensitive, so they can be killed by even different concentration of other surfactant that might be more gentle. So it's really difficult. That then have to be tested every time with CACO2 cells. So and with the Pampa, there are several problems with Tritonex, Solutol, and others. So it's
very important to do the work before to start your experiment.
Yeah, you have to.
Yeah. Okay, the next question comes from Dominic.
Yeah.
Another disadvantage of cellular-based assays in the trapping of drug in the cells, leading to the typical reported recovery in percentage. Is there already an understanding of how much drug potentially remains in the lipid layer of PermePet?
We have made some estimation for hydrophilic compounds. It's very low, basically zero. For lipophilic compounds, we tested hydrocortisone, and we got, I think, a maximum entrapment we got that was around 5%, so not much.
Isn't there a chance to test the concentration of drug in the lipid layer if you use front cells or side-by-side cells by dissolute the lipid layer afterwards?
Yeah, that's it. For the PermePad, you mean?
Yes.
Yeah, that's actually what we do generally. We dissolve the barrier, the lipid layer inside the organic solvent, and then we measure it with HPLC to know how much drug has remained in the lipid layer.
Yeah, but this is for PermePet in the 96 well plate. This is unfortunately not doable.
Or maybe you can try to do it also, but it's more complicated.
Yeah, with the glue is quite difficult.
Yeah.
Okay, the next question. I hope it answered it a bit, Dominic. How much accurate permeability prediction for BBB using PermePet in case study?
This is very interesting. We don't, as far as I know, there is not big comparison between BBB and the PermePet. PermePet has been designed for intestinal, and also it's very good in predicting, as I said, small thin mucosal layer, nasal mucosa, buccal. It has never been compared to BBB, but we have to remember that BBB is a very different type of barrier. It's extremely tight, doesn't express this tight junction, and also the absorption is really related to active transport. So for the BBB, I don't think PermePed is actually a proper model, but it's a model to understand the passive permeation of the drug that you want to deliver it to the BVA. So to say, if you get a very high permeation, you can expect permeation for the blood-brain barrier. But in this case, I would definitely focus on cellular models, or yeah.
Okay.
The BBB is peculiar.
Good. The next question is, can these synthetic membranes be used to test permeation over 24 hours? Can they maintain their integrity throughout?
Yes, they can.
There are several integrity tests out there to see how stable it is. I can say for PermaPad there is an insurance to test up to 24 hours, and we have customers which are using them even longer. Okay, now a question from Aditya. Could you share publications white paper, if available, of using PermaPad with Pion macroflux system?
I think you should answer, Yannick, to this one.
Yes. Right now, I don't have any data or publication out there using the macroflux system. We have some test customers who are using these cartridges, but this is not published anywhere. It's just a test phase. I'm sorry. Okay, the next question from Dominic. Which organic solvent is considered suitable to dissolve the PermaPad membrane extract drug from the PermaPad membrane? PS.
What I utilized, we utilized methanol. Methanol is very good, but also ethanol to some extent. But methanol is I would choose methanol actually. Thank you, Dominic, for your interesting questions. Yeah.
I got another question on the private channel. If there is a possibility to discuss a special application with special formulation in a separate meeting.
Yes, that's absolutely fine to me. Of course, maybe we can discuss it with Yannick and separate. But absolutely, yes, no problem for me.
So if you are interested in a separate meeting, you can just contact us at info@permapad.com, so you can reach us. I just type it in the chat.
Yes.
Any other questions? If not, I would like to make a little marketing to make sure for you to follow our LinkedIn page, Fabiok, to stay updated and get the newest news about everything, about new publications, about the Permeability series, and I think it's a very good channel to stay updated. You're welcome, Amparo. The most work was done for Max, of course. Okay, so if there is no question left, if you have questions afterwards, going through the presentation again, please feel free to contact us anytime. And we will stay in touch and see you at the next Permeability series webinar. Have a nice afternoon, evening, or night. I saw someone from Japan. It's quite late there, so and India, of course. Have a good day and talk to you soon. Bye bye.
Bye bye. Thank you.